A grid cell is a type of neuron in the brains of many species that allows them to understand their position in space. Grid cells derive their name from the fact that connecting the centers of their firing fields gives a triangular grid. Grid cells were discovered in 2005 by Edvard Moser, May-Britt Moser, and their students Torkel Hafting, Marianne Fyhn and Stella Molden at the Center for the Biology of Memory, CBM, in Norway. They were awarded the 2014 Nobel Prize in Physiology or Medicine together with John O'Keefe for their discoveries of cells that constitute a positioning system in the brain. The arrangement of spatial firing fields all at equal distances from their neighbors led to a hypothesis that these cells encode a cognitive representation of Euclidean space. The discovery also suggested a mechanism for dynamic computation of self-position based on continuously updated information about position and direction. In a typical experimental study, an electrode capable of recording the activity of an individual neuron is implanted in the cerebral cortex of a rat, in a section called the dorsomedial entorhinal cortex, and recordings are made as the rat moves around freely in an open arena. For a grid cell, if a dot is placed at the location of the rat's head every time the neuron emits an action potential, then as illustrated in the adjoining figure, these dots build up over time to form a set of small clusters, and the clusters form the vertices of a grid of equilateral triangles. This regular triangle pattern is what distinguishes grid cells from other types of cells that show spatial firing. By contrast, if a place cell from the rat hippocampus is examined in the same way, i.e., by placing a dot at the location of the rat's head whenever the cell emits an action potential, then the dots build up to form small clusters, but frequently there is only one cluster, one place field, in a given environment, and even when multiple clusters are seen, there is no perceptible regularity in their arrangement. Background. In 1971, John O'Keefe and Jonathan Dustrovsky reported the discovery of place cells in the rat hippocampus cells that fire action potentials when an animal passes through a specific small region of space, which is called the place field of the cell. This discovery, although controversial at first, led to a series of investigations that culminated in the 1978 publication of a book by O'Keefe and his colleague Lynn Nadel called The Hippocampus as a Cognitive Map. The book argued that the hippocampal neural network instantiates cognitive maps as hypothesized by the psychologist Edward C. Tolman. This theory aroused a great deal of interest, and motivated hundreds of experimental studies aimed at clarifying the role of the hippocampus in spatial memory and spatial navigation. Because the entorhinal cortex provides by far the largest input to the hippocampus, it was clearly important to understand the spatial firing properties of entorhinal neurons. The earliest studies, such as Quirk et al., 1992, described neurons in the entorhinal cortex as having relatively large and fuzzy place fields. The Mosers, however, thought it was possible that a different result would be obtained if recordings were made from a different part of the entorhinal cortex. The entorhinal cortex is a strip of tissue running along the back edge of the rat brain from the ventral to the dorsal sides. Anatomical studies had shown that different sectors of the entorhinal cortex project to different levels of the hippocampus. The dorsal end of the EC projects to the dorsal hippocampus, the ventral end to the ventral hippocampus. This was relevant because several studies had shown that place cells in the dorsal hippocampus have considerably sharper place fields than cells from more ventral levels. Every study of entorhinal spatial activity prior to 2004, however, had made use of electrodes implanted near the ventral end of the EC accordingly. Together with Marianne Fyhn, Stella Molden, and Menno Witter, the Mosers set out to examine spatial firing from the different dorsal to ventral levels of the entorhinal cortex. They found that in the dorsal part of medial entorhinal cortex, MEC, cells had sharply defined place fields like in the hippocampus, but the cells fired at multiple locations. The arrangement of the firing fields showed hints of regularity, but the size of the environment was too small for spatial periodicity to be visible in this study. The next set of experiments, reported in 2005, made use of a larger environment, which led to the recognition that the cells were actually firing in a hexagonal grid pattern. The study showed that cells at similar dorsal to ventral mech levels had similar grid spacing and grid orientation, but the phase of the grid, the offset of the grid vertices relative to the x and y axes, appeared to be randomly distributed between cells. The periodic firing pattern was expressed independently of the configuration of landmarks, in darkness as well as in the presence of visible landmarks, and independently of changes in the animal's speed and direction, leading the authors to suggest that grid cells expressed a path integration-dependent dynamic computation of the animal's location. For their discovery of grid cells, May-Britt Moser and Edvard Moser were awarded the Nobel Prize in Physiology or Medicine in 2014 alongside John O'Keefe. Properties. Grid cells are neurons that fire when a freely moving animal traverses a set of small regions, firing fields, which are roughly equal in size and arranged in a periodic triangular array that covers the entire available environment. Cells with this firing pattern have been found in all layers of the dorsocaudal medial entorhinal cortex, DMEC, but cells in different layers tend to differ in other respects. Layer 2 contains the largest density of pure grid cells, in the sense that they fire equally regardless of the direction in which an animal traverses a grid location. Grid cells from deeper layers are intermingled with conjunctive cells and head direction cells, i.e. in layers 3, V and 6 there are cells with a grid-like pattern that fire only when the animal is facing a particular direction. Grid cells that lie next to one another, i.e. cells recorded from the same electrode, usually show the same grid spacing and orientation, but their grid vertices are displaced from one another by apparently random offsets. Cells recorded from separate electrodes at a distance from one another, however, frequently show different grid spacings. Cells that are located more ventrally, that is, farther from the dorsal border of the mech, generally have larger firing fields at each grid vertex, and correspondingly greater spacing between the grid vertices. The total range of grid spacings is not well established. The initial report described a roughly twofold range of grid spacings, from 39 cm to 73 cm, across the dorsal most part, upper 25%, of the mech, but there are indications of considerably larger grid scales in more ventral zones. Brun et al., 2008, recorded grid cells from multiple levels in rats running along an 18-meter track, and found that the grid spacing expanded from about 25 cm in their dorsalmost sites to about 3 m at the ventralmost sites. These recordings only extended 3/4 of the way to the ventral tip, so it is possible that even larger grids exist. Interestingly, such multi-scale representations have been shown to be information theoretically desirable. Grid cell activity does not require visual input, since grid patterns remain unchanged when all the lights in an environment are turned off. When visual cues are present, however, they exert strong control over the alignment of the grids. Rotating a cue card on the wall of a cylinder causes grid patterns to rotate by the same amount. Grid patterns appear on the first entrance of an animal into a novel environment, and usually remain stable thereafter. When an animal is moved into a completely different environment, grid cells maintain their grid spacing, and the grids of neighboring cells maintain their relative offsets. Interactions with hippocampal place cells. When a rat is moved to a different environment, the spatial activity patterns of hippocampal place cells usually show complete remapping, that is, the pattern of place fields reorganizes in a way that bears no detectable resemblance to the pattern in the original environment. Muller and Kubi, 1987. If the features of an environment are altered less radically, however, the place field pattern may show a lesser degree of change, referred to as rate remapping, in which many cells alter their firing rates but the majority of cells retain place fields in the same locations as before. Fhyhnetal, 2007, examined this phenomenon using simultaneous recordings of hippocampal and entorhinal cells, and found that in situations where the hippocampus shows rate remapping, grid cells show unaltered firing patterns, whereas when the hippocampus shows complete remapping, grid cell firing patterns show unpredictable shifts and rotations. Theta rhythmicity. Neural activity in nearly every part of the hippocampal system is modulated by the limbic theta rhythm, which has a frequency range of about 6-9 Hz in rats. The entorhinal cortex is no exception. Like the hippocampus, it receives cholinergic and GABergic input from the medial septal area, the central controller of theta. Grid cells, like hippocampal place cells, show strong theta modulation. Grid cells from layer 2 of the mech also resemble hippocampal place cells in that they show phase precession, that is, their spike activity advances from late to early phases of the theta cycle as an animal passes through a grid vertex. Most grid cells from layer 3 do not precess, but their spike activity is largely confined to half of the theta cycle. The grid cell phase precession is not derived from the hippocampus, because it continues to appear in animals whose hippocampus has been inactivated by an agonist of GABA. Possible functions. Many species of mammals can keep track of spatial location even in the absence of visual, auditory, olfactory, or tactile cues. By integrating their movements, the ability to do this is referred to in the literature as path integration. A number of theoretical models have explored mechanisms by which path integration could be performed by neural networks. In most models, such as those of Samsonovich and McNaughton, 1997, or Burek and Fayet, 2009, the principal ingredients are, 1. An internal representation of position, 2. Internal representations of the speed and direction of movement, and 3. A mechanism for shifting the encoded position by the right amount when the animal moves. Because cells in the mech encode information about position, grid cells, and movement, head direction cells and conjunctive position bidirection cells, this area is currently viewed as the most promising candidate for the place in the brain where path integration occurs. However, the question remains unresolved, as in humans the entorhinal cortex does not appear to be required for path integration. Burek and Fayet, 2009, showed that a computational simulation of the grid cell system was capable of performing path integration to a high level of accuracy. However, more recent theoretical work has suggested that grid cells might perform a more general denoising process not necessarily related to spatial processing. Hafting et al., 2005, suggested that a place code is computed in the entorhinal cortex and fed into the hippocampus, which may make the associations between place and events that are needed for the formation of memories. In contrast to a hippocampal place cell, a grid cell has multiple firing fields, with regular spacing, which tessellate the environment in a hexagonal pattern. The unique properties of grid cells are as follows. Grid cells have firing fields dispersed over the entire environment, in contrast to place fields which are restricted to certain specific regions of the environment. The firing fields are organized into a hexagonal lattice. Firing fields are generally equally spaced apart, such that the distance from one firing field to all 6 adjacent firing fields is approximately the same, though when an environment is resized, the field spacing may shrink or expand differently in different directions. Berry et al. 2007. Firing fields are equally positioned, such that the 6 neighboring fields are located at approximately 60-degree increments. The grid cells are anchored to external landmarks, but persist in darkness, suggesting that grid cells may be part of a self-motion-based map of the spatial environment.
Please subscribe and thanks for watching.